Welcome to this explainer. Today, we're unpacking a massive biological plot twist about how sugar actually interacts with our bodies. We're going to look at two entirely different biological environments. You see, the systemic sugar danger we fear the absolute most, it's largely a myth. But the danger we completely ignore, it's happening right inside our mouths as we speak. We're taking a journey from the macro environment of cancer metabolism all the way down to the highly localized microenvironment of your teeth. Let's get into it.
So, does dietary sugar directly feed cancer? Let's dive right in and tackle one of the most widespread, anxiety-inducing questions in modern nutrition. I mean, if you spend 5 minutes on the internet, you'll absolutely find warnings screaming that eating sugar directly causes tumors to grow. And this belief has seriously influenced public perception. It causes a lot of cancer patients to adopt highly restrictive diets, often when they're already super vulnerable to malnutrition and weight loss. But what does the actual biological and epidemiological evidence say about this direct link?
The idea that the sugar on your plate directly fuels cancer actually stems from a massive misinterpretation of a biological phenomenon known as the Warburg effect. Discovered by Otto Warburg, this effect basically describes how many malignant cells drastically alter their metabolism. They heavily increase their glucose uptake and convert it into lactate, even when they're sitting in plenty of oxygen. But here is the absolute crucial misinterpretation. Just because cancer cells reprogram themselves to gorge on glucose from your bloodstream, it doesn't mean that the sugar you eat is the direct trigger for that reprogramming. And it definitely doesn't mean that avoiding dietary sugar will magically starve the tumor.
Let's look at the stark contrast in energy efficiency here. Normal cells, they're like highly efficient hybrid cars. They use this beautiful multi-step process involving the mitochondria to extract an impressive 36 to 38 molecules of ATP, the energy currency of the cell, from just a single molecule of glucose. But cancer cells? They bypass this efficient route entirely. They only get 2 ATP per glucose molecule. 2! So why on earth would they do this? Well, they're intentionally trading energy efficiency for the ability to rapidly build new cells. They're literally using the byproducts of this super inefficient process as the building blocks for new DNA, proteins, and lipids.
And this brilliantly illustrates exactly why you can't just starve a tumor by cutting carbs out of your diet. Human biology is incredibly resilient. It's designed to maintain steady blood glucose levels even if you stop eating sugar entirely. Your liver will literally just manufacture glucose for you. But okay, even if glucose levels somehow drop in the tumor microenvironment, these cancer cells have incredible metabolic plasticity. They're flexible engines. If they can't get glucose, no problem. They just reroute their systems to run on alternative carbon sources, like the amino acid glutamine or fatty acids. This crazy adaptability is exactly what makes targeting cancer metabolism so incredibly complex.
Now, you might be sitting there thinking, wait a minute, what about those glowing PET scans? Don't they literally show tumors eating up radioactive sugar? It's a totally fair question and a fascinating piece of the puzzle. Those FDG PET scans absolutely do highlight areas of high glucose uptake. But recent dual tracer studies have revealed something wild. Those bright glowing hotspots, they aren't the cancer cells gorging on sugar at all. Plot twist, they're actually your own immune cells. Yep, specifically macrophages and T cells, working absolute overtime in the tumor microenvironment to fight the cancer. Meanwhile, the actual malignant cells might be busy munching on glutamine instead.
So, the crucial takeaway here is that when we look at massive population studies across various cancers, like breast, endometrial, colorectal, and ovarian, the epidemiological evidence linking them directly to dietary sugar is overwhelmingly no. Seriously, mostly null across the board unless we factor in something else. When large prospective cohorts actually do find a link, those positive associations are almost entirely confined to metabolically susceptible groups. And get this, the link basically disappears once we adjust for factors like total energy intake and body mass index.
Okay, so how does sugar actually matter in this context? Well, it works indirectly. Let's walk through this sequence. When excess sugar intake leads to a chronic energy surplus, it promotes adiposity, excess body fat. And we know this fat isn't just sitting there inertly. It creates insulin resistance, which leads to chronically high insulin levels and elevates growth pathways like IGF-1. Plus, it promotes low-grade systemic inflammation. So in this way, chronic metabolic dysfunction acts a lot like a fertilizer for a pro-tumorigenic environment. It's setting the stage rather than dietary sugar acting as a direct fuel injected right into the tumor.
Having completely debunked the macro myth of sugar directly feeding systemic cancer, we're going to pivot. We're moving from that massive systemic metabolic environment to a very real micro-level biological battle happening right now in your mouth. Because while sugar is really only an indirect player in cancer, it is the direct absolute fuel for one of the most prevalent infectious diseases in the entire world, dental caries, or as we usually call them, cavities.
Meet the actual villain of our sugar story, a bacterium called Streptococcus mutans. Now, on its own, it's just another microbe hanging out in your oral microbiome. No big deal. But when it encounters the dietary sucrose you eat, it essentially acts like a microscopic supervillain grabbing its ultimate weapon. It weaponizes that sugar using a highly specialized set of enzymes called glucosyltransferases. We'll just call them GTF enzymes to keep things simple. This specific interaction right here is the absolute initiation point for tooth decay.
And you know, this isn't just some simple story of bacteria eating sugar and sweating out acid. It's way more sinister. The S. mutans bacteria are literally using the sugar you consume as biological bricks. And those GTF enzymes we just mentioned, that's the mortar. They use them to synthesize these sticky, water-insoluble extracellular polysaccharides, or EPS. Basically, they're building a dense, 3D biofilm fortress right on the surface of your tooth. Once they're inside this fortress, the bacteria are perfectly protected from harsh environments, from your own immune system, and even from traditional therapies. They just sit in there, undisturbed, producing acid and destroying your tooth enamel. Now, for decades, our approach to fighting this has basically been like dropping a bomb on the entire oral ecosystem. Traditional chemical mouthwashes and broad-spectrum antibiotics, they don't discriminate. They kill both the bad bacteria and the highly beneficial commensal bacteria. This completely ruins your natural oral microbiome, and worse, it can drive antibiotic resistance. So scientists have been desperately searching for a targeted saboteur instead. They wanted a non-bactericidal agent, something that could precisely disarm that harmful biofilm machinery without wiping out the good microbes your mouth actually needs to stay perfectly balanced.
To find this targeted saboteur, researchers ran a massive screening of over 1,000 natural compounds. And in this incredible breakthrough, they found a totally surprising hero: tannic acid. Yeah, tannic acid. It's a natural, complex polyphenol that you'll find in everyday plants like pomegranates, gallnuts, and chestnut leaves. Out of all the thousands of compounds they tested, tannic acid emerged as uniquely capable of specifically targeting and shutting down that crucial GTF enzyme we talked about. Check out this specific number: negative 11.3 kilocalories per mole. That represents the molecular docking binding energy between tannic acid and the GTF protein. Now, you absolutely do not need to be a biochemist to appreciate this. All you need to know is that in the chemistry world, a highly negative binding energy means a remarkably strong affinity. It's an incredibly tight lock. This proves, both mathematically and through cool 3D structural mapping, that tannic acid binds fiercely to the problematic GTF enzyme. It literally locks right onto the machinery and shuts the whole operation down.
What makes tannic acid so utterly brilliant is exactly how it operates. It selectively antagonizes the GTF enzyme, which totally halts the synthesis of that sticky EPS, completely stopping the biofilm fortress from ever forming. But here's the real magic. It does all of this without actually killing the S. mutant bacteria directly. By simply disarming the pathogen rather than destroying it, tannic acid prevents the nasty plaque buildup, avoids driving any kind of antibiotic resistance, and perfectly maintains that healthy physiological balance we want in the oral microbiome. It's a total game changer.
As we wrap up this explainer, I want to leave you with a final thought. Science shows us that our relationship with sugar is way more nuanced than the simple cause and effect myths we read online. It's not this direct magical fuel for cancer, but rather a contributor to systemic metabolic dysfunction when we consume it in major excess. And in our mouths, it requires targeted biological interventions, not just broad-spectrum destruction. So what if, instead of being blindly terrified of sugar causing cancer, we shifted our focus? What if we channeled that energy into maintaining systemic metabolic balance and protecting the incredible microscopic ecosystems right inside our mouths?